Okay. ¿Sigo compartiendo pantalla completa? No.
Sí.
Ahí está, ya.
Sí, un segundo y ya estamos. Nada más.
Sí.
Listo. Ok.
Muy bien, vamos a comenzar entonces. Ready.
Good morning. If you can take your seat, please, we're going to start with our next conference. Well, our next speaker is going to be Dr. Hugo Esquivel. He's a veterinarian, a Master Science degree, and a PhD in Biomedical Science from UNAM. He is principal investigator at the Medical and Pharmaceutical Biotechnology Unit of the Centro de Investigación y Asistencia en Tecnología y Diseño del Estado de Jalisco, in Guadalajara. He has directed several scientific and technological projects sponsored by private and public funds. Founder of the research topic of biomedical engineering, of biotechnological drug and tissue engineering, and the preclinical studies unit, and responsible for the laboratory of regenerative medicine and tissue engineering in NCIATEA. Member of the National System of Researchers with Level 1 distinction of CONACYT's accredited evaluator and peer review referee of many scientific international journals. He has tutored several masters and doctors, author of 21 international scientific papers and 8 patents. His principal aim are the research and development of novel therapies against human and bovine tuberculosis and for the main lethal diseases affecting the vulnerable Mexican population. So please welcome Dr. Hugo Esquivel with a round of applause.
Thank you very much for the kind introduction. Can you hear me well?
Yes, we can hear you.
Thank you very much. Well, thanks again for the kind invitation. My brief title of my talk is interleukin gene therapy in a mouse model of liver cirrhosis. I would like to thank the organizers committee for the kind invitation to this amazing and amusement STEM week at university. Well, my talk will be divided in several topics. First of all, I would like to talk about the huge problem we have as a society, then the possible solution that we approach and the experimental that we've done, and some of the results that we already have in the lab, some conclusions, and I will be able to respond some of your questions. Well, as you know, my talk is focused on the liver. This is the kind of controversial organ that as human and mammals we have, because it can be the factor for evolution of the species, but also the factor for the lethal outcome when we have disease in this organ. First of all, the liver is located anatomically in the stomach, well, in the cavity of the stomach, and is beneath of the diaphragm, and is so huge that it can weigh up to 2 kilograms in a regular adult. So when the liver becomes the central part of my talk, I mean it because it's the specialized organ that works, functioning, doing that any other organ can do. First, for example, this organ is responsible for cleaning our blood. It can filters almost 2 liters per second to retry all the waste that the other tissues do, and for cleans also from drugs, for instance, and for the metabolites of regular metabolism from proteins, sugars, and also fats. So it is important cleaning our blood. The second function, or the second transcendent work that the livers do, is to produce the bile, that is a liquid important for digestion, that emulsificate the fats that we acquire from the food in order to make them small, in order to get better digestion, for taking approach of these small lipid droplets that will be used to build more fat or even energy. The third function of the liver is really involved in the metabolism of nutrients. For example, the glucose. Glucose will be transformed in the liver in a bigger molecule to store that will be not used regularly, or will be used later on when you are fasting. This bigger molecule made by glucose is called glycogen. So the liver is the first store, or the first reserve of glycogen in the body. The second one is the muscle, the skeletal muscle. So the liver is also important storing or taking abroad the nutrients that will not be used immediately. The fourth function, the fourth relevant function of the liver is the synthesis of proteins that will be delivered to the blood plasma. These proteins are really important to sustain the blood pressure and to carry on other materials, for example, the drugs that we take to combat any illness or any disease, and to metabolize them inside the liver. The five no less important function is to produce or even store vitamins and minerals that we're no able acquire by the nutrients, the regular food, or any other sources. But the liver itself has more than 500 or even more functions that any other organ can do. So the liver is quite important to sustain our life, quite important to maintain our health, systemic health, and it's involved in several diseases when it comes ill. So it's really important to have a healthy liver to be aware of those diseases and to maintain our regular activities without taking account of this very important organ. So when the liver starts to fail or starts to ill because we don't have regular or healthy habits, then it begins the problem. And our liver is capable to restore itself until it no longer can and ask us for help. When it happens, the liver has already damaged almost beyond the 30% and almost the 60%. It means that when one starts to show clinical symptoms related to a liver disease, is because the liver is already ill and there's no much room for to attend this disease, and no much margin to solve the problem. So it becomes a really huge problem. What is the liver disease? Well, in this image, up you can see a more or less healthy liver that is brown, red, or wine red, and below is the liver, the very damaged liver that becomes fibrotic, unfunctional, and needs to be replaced. It means you or someone that has a liver with this aspect has a liver with cirrhosis. What is cirrhosis? Well, it's the end stage of a liver disease that begins from a liver healthy at all, that it starts to accumulate tiny droplets of lipids coming from the bloodstream that are acquired to store it, because somehow the liver starts to manipulate this fat to become energy storage or to dry them from the bloodstream. It means that the liver becomes a sacrifice organ to maintain the healthy organisms from several offenses, several stresses that we don't notice until the liver progresses to a more advanced disease. It means the liver becomes fibrotic. What does it mean, a liver becoming fibrotic? If you lesion your skin, you will be bleeding immediately, that it serves for the source of cells that will repair this lesion. Between these cells are immunological cells, or cells coming from the immune systems, that are attaching to the tissue in order to keep two borders bind and to deliver new factors that will replace the tissue that has already gone. But also, there will be other kind of cells that will produce extracellular matrix, mainly some kind of proteins that has the ability to strongly attach each border. Those are called collagens or fibrins, in order to maintain the surface, intact surface of the tissue. The same happens to a liver when it is lesioned by different kind of stressors. For example, would you take an aspirin, or when you take an ibuprofen, or when you take another ain in order to get your headache controlled or abated, this medicine, this drug, will be metabolized by the liver. When the liver is healthy, not much happens. The medicine or the drug is metabolized, it's drained off from your body, and you can continue your life with no any situation. But when the liver has a first lesion, for example, you came after a Friday party and took 2, 3, 4, 5, 6 drinks, and you want to cure your hang-up, you take those medicines without taking account that your liver is already stressed. If you do this frequently, for example, each weekend during your stay at the university, and then the liver becomes ill, and you don't notice, you don't see anything happening to your body, you don't see anything happening to your skin, you don't see anything happening to your constant, even if you take a blood analysis, you won't see it. So this is why the liver disease is often a chronic disease. It means that before you notice that your liver is already damaged, you previously have passed the mild or even damage that could be seen in a regular laboratory exam, until the liver no longer can sustain this stress. So the stage of cirrhosis means that the liver has already accumulated a long enough lesions from different sources that the liver can no longer sustain. In this stage, the liver has tried to repair even lesion you or anyone has done to its own organ, but no longer can repair its lesions. It needs more cells, it needs more help from the body, but it no longer receives that. So this is sad because the majority of cases in the clinics starts a diagnosis in this stage or in the previous one when the liver has enough fibrosis that the clinical cannot do much for the patient. So I would like you to present this disease as the real problem we have, because the next data I will provide you is, for me, alarming and needs some attention before we have these stages of liver damage. So this is a real liver with a deadly end stage of liver disease, full of cirrhosis and some nodules with liver cancer. The second cause of death of cancer by all the types of cancer in Mexico in males, and the fifth in females. So this is a huge deadly problem to me, and hope at the end of this talk will be for you too. Well, what have in common those states from the US, New Mexico, Nebraska, Idaho, and those states from Mexico, Morelos, Quintana Roo, and Durango? All of them have more or less 2 million people. This is what 1 million people looks like, all gathered in the same place. Well, this is twice the number of people dying by this deadly disease each year in the adult population that will be working, having a good life, and more expectations. But sadly, it shouldn't. What are the first etiologies associated with these deaths by cirrhosis, meaning liver disease? The first two are caused by viruses. One, the most deadly, is the hepatitis C virus. The second one is the hepatitis B. But surprisingly or not surprisingly, it depends how do you see it, alcohol consumption is the third cause of liver cirrhosis and the third killer from the human populations about this disease. The third one, taking in the viruses as just one, the third one, it is a disease that it's increasing, and nowadays is a global pandemic called non-alcoholic fatty liver disease. That actually the deadly end of this disease is non-alcoholic steatohepatitis. It means that the liver, it becomes more than fibrotic, but also it becomes to progress with aggressive inflammation to other organs, affecting, for example, the heart, the brain, the kidneys, and so on. So this is a deadly disease that in the beginning is silent. It is not noticed, but when you have NASH going through NAF, then you have to act quickly in order to preserve your life. So what are I'm sorry. Sorry, I don't know what, okay. Well, okay. What are the data about this disease around the world? Well, as I mentioned, this is a deadly disease. It accounts for 4% of all deaths worldwide. It means that if you can count 50 mates in your room, or even 100, 4 of your mates will be dead if they don't take account of this information from liver disease. Yeah, I know it's funny when you hear it, but it's real. It's real. For example, 2 out of 3 liver-related deaths occurs among men, among males, more than in females, than in women. So globally, this disease accounts just more than 2.4% of global deaths. It means that if you have counted 50 people in that room, maybe 1 or more will die for this disease, and you will not notice until it's too late. This is the image of Mexico around the world. You can see the incidence of deaths from this cirrhotic liver disease that takes more than 300,000 people for just this pathology in our country. No matter if it is a man or if it is a woman. So cirrhosis by itself, just as a single cause of death from all the growth of liver disease, is the 10th leading cause of death in Africa, the 9th in Southeast Asia and Europe, and the 5th leading cause of death in the Eastern Mediterranean. So again, our data are not so far from those data I already presented. We don't have accurate data, sadly to say, because in Mexico, liver disease is accounted just as a one single pathology, becoming from the fatty liver disease to the non-alcoholic steatohepatitis. So for the diagnosis of death, one will die for this disease, but not the reason if have steatohepatitis or just fatty liver disease. But what is the first etiology for the Mexican population dying from this disease? Is the alcohol consumption. So as you can see in this picture, in the left, you can see the rates of men to women dying for this disease is almost in the middle of the age of young and elder men in Mexico, almost 5% of death in our country just due to this lethal disease. In the right, you will see the prognostic, the prediction of this disease to 2019. Well, you can see the race more or beyond the 6% of the deaths just for this disease by alcohol consumption. The second cause of this illness, remember, is avoiding the viral pathology. I just meaning the not infectious diseases, but more regulated by lifestyle, for example, alcohol consumption in the first one. The second one is overweight and obesity. Those treated as just one is the second big one cause of this lethal disease. Concurring also, and obviously, with diabetes, type 2 diabetes, that also increases by a 12% the cases of death of this disease in Mexico, just in Mexico. So, is any treatment for this sad, chronic, and deadly disease? Well, the news is not. the mid treatment for cirrhosis. It means that when the liver has become fibrotic and with the almost no return point, just the recommendation is to slow to recover by itself, give them our cells the margin to recover, to repair the liver, is to slow the liver damage or the liver transplant. But if you have started to develop nodules of cancer, you are no longer a candidate for liver transplant. So you are condemned. Yeah, I know. I'm so rude, but it is the intention. I need you to understand that it's a deadly disease. So, and you are in the middle of the edge of the population that is coming the focus of this illness. So let's talk about interleukin 10. Interleukin 10 is a cytokine produced mainly by cells from the immune system. Probably you have read about it because the previous pandemic of that was caused by a virus. So this cytokine is produced to counteract the pro-inflammatory effect of the immune system. It means that this interleukin is anti-inflammatory. It means that will defeat or will reverse all that the inflammation at the beginning of an infection or a lesion started. So, but interestingly, the hepatocytes that are no regular or known cells from the immune system, hepatocytes are the singular functional cells from the liver that do what all the functions of the liver does. The hepatocytes by itself produce interleukin 10 as a modulatory effect on liver fibrosis. This is the first signal that the liver has to counteract the effects of the inflammation, the chronic inflammation that has overcome the point of no return, turning the liver in a like a stone, unfunctional, fibrotic organ. So interleukin is produced in the liver, trying to counteract this pathology. But it cannot solve all the damage one has done to this organ. So maybe if we help the amount that the liver cannot produce by external supply, maybe we can overcome this deficiency of our organism when it's stressed to the top. And maybe by gene therapy, it means supplying an external gene to be expressed, and the protein produced by this gene, naming interleukin 10, will overcome or surpass this point. What other scientists have seen is that supplying gene therapy has prevented cell apoptosis, that is a type of cell death in the liver, and to reverse some kind of the fibrotic process in livers that have been treated with a fibrotic drug called thiacetamide. So the interleukin 10 could reverse the damage that the liver cannot do by itself by providing external supply. However, the drawbacks of to provide external interleukin 10, for instance, recombinant protein, are the elevated or the high levels of interleukin 10 than when you deliver a single shot. Because the secondary effects, not health secondary effects, it can suppress your host response to regular microbes, becoming them pathogenic. So it is not a good idea to block your regular immune system by providing huge amounts of these anti-inflammatory cytokine. The second one is that it's so good to alleviating or to preventing these drawbacks from the first inflammatory stages that also prevents the resolution of the associated tissue damage. It means that it's so elevated the levels, the doses, that you cannot control when you give a shot of this interleukin, that cannot you see more damage than solution. And also you can see disturbances in your blood, for example, in the number of the cells that are regulated by this cytokine. So it's not a good idea to use as external therapy to this deadly disease. So what is the solution? Well, maybe if we can afford a very effective treatment supplying external interleukin-10 through a gene therapy, but that will be tissue specific and self-regulated. It means self-regulated with a switch on when it is necessary and a switch off when no longer is needed. Well, what was our experimental approach? We designed an expression vector having two regions that were synthetic in the entire that function as this switch on-off, and a promoter that will be just switch on when it is done by a cell from the liver, namely an hepatocyte or namely stellate liver cell that are only present on this organ. So then we built different versions of this vector in order to test each singular switch and to test mainly by expressing gene fluorescent protein, not interleukin-10, until we finally found the right combination to test this hypothesis, this expression vector. So what did we do to test this hypothesis, if this vector already worked? Well, first of all, we test the system to switch on and switch off. And we transfect two different type of cells. One is specific for the liver, that is hepatic stellate cells from isolated from a sample from a biopsy of a human liver, healthy human liver. And another one that is regular use to transfect cells in order to express recombinant proteins. It is not associated from the liver. Actually, it is a kidney cells from a monkey. So we use those two type of cells just to try if with the signals that we are providing to these cells could serve as starters or as stimulants to the production of this gene product. This gene product in this stage was GF protein. So as you can see, we measure the red fluorescent signal on each cell, and we take the counts and we compare them. So as we can see, in the red, let me try to, okay. Those two bars correspond to the switch on and switch off parts of the vector that were compared to another construct that has no switch on and switch off, but has a strong enhancer of the expression that is part of the construct that becomes from a virus that infects any kind of cell. So in this part of the development of the testing, we try to compare the intensity of the expression regulated by the inflammatory stimulus. LPS is a part of a bacteria, a gram-negative bacteria, that it's pro-inflammatory by itself. And this molecule exerts an hypoxic environment just by chemistry, binding by the hypoxic transcriptor factor called HIF1, and we can see that with those stimulus, we see a raise above the regular one that has no control, meaning that it has a strong response, and compared with those that, even though has the same stimulus, doesn't have the switch on, switch off part of the DNS. So at the cell level, we can test that it worked. So it corresponds to these two constructs. Then we move off to the in vitro tissue model of this fibrotic process. We can see in red the protein called small muscle actin alpha that is just produced by myofibroblasts that become fibrotic and are part of the process. And we measure the same signal, the GFP, but by intensity, not by each cell, but by intensity. And we observe the same as the previous one, comparing the treatment, the hypoxic treatment, without treat. So we can confirm that in a model of fibrotic process, you can have a huge transduction and a huge production of the GFP protein. And we confirm that this system under hypoxic conditions were modulated by this transcription factor. Then we move on to the next test in the specific tissue. We use several hepatocytes cell lines coming from human and compared to that one of mouse and to other kind of cells that don't belong to the liver, and we can confirm that were liver specific. For the promoter, we included in this stage of the development, and we can confirm that they were overexpressed the genes that are already in the normal liver. So then we next move to the next stage, that was the gold standard test to see if there is an effect therapeutic, taking into account that we have proof that the model of the vector starts and finishes the production of the protein, GFP protein, by those signals that are present in the fibrotic process, and also that is only specific for the liver, cells for the liver, and then move on to the therapeutic effect. We use a model that has hepatocytes in the top and stellate cells in the bottom, inside a well of a cell culture plate. And then we stressed the hepatocytes that we transfect. And you can see that when we submit to these different wells to the same stimulus, like hypoxia, and then we supply the vector, the therapy under the same environment, you can see the reduction of biomarkers of fibrotic process, and also at the level of gene expression. For instance, the previous one that I already mentioned, the small muscle actin, you can see a reduction due to this therapy. So we can move on to the in vivo test. So we induce by CCL4 treatment, that is very lethal when you overdose this treatment in the mice, to develop fibrosis for 42 days. And then we start our treatment for several days. So we started with already mice with liver cirrhosis. And we performed just three treatments, because we wanted to assure that the expression was sustained and it was not exposed for the cells, because you are injecting an external DNA. And then we made the analysis. In this movie, you can see how the therapy, 4 milliliters, are supplied through the tail vein to the mice, for each single mice having the cirrhotic process. And also we compare to mice healthy and with mice that only receive the vehicle called saline. And this image, we can confirm that in the majority, the vector was allocated in the liver, no, in this signal for the GFP image, and some of the vector also reached another organs of the mice with no complication. So in the analysis, we compare those mice that were cirrhotic with those mice that were treated with the on complete vector, and those mice that were treated with complete vector versus the control, healthy control. And in those images, you can see that there is a slight modification of the architecture of the liver, but in this image, this row represents the accumulation of glycogen. The more violet you see the image, the more glycogen is stored. So you can see that in the mice that has the cirrhotic process, without the treatment, there is almost no store of glycogen, meaning that this liver no longer works. And also we measure the survival of this, each mouse on each group. And this line represents the group that was not treated after 14 weeks of study. All the mice died at the week 16, and all those that we treated with our system, partially complete or full system, all survived to the end of the study. And we test that the interleukin-10 levels on the bloodstream. This is the Western blot of plasma of some of the mice that were bleed along the study. And you can see the overexpression or overcontent of interleukin-10 homonomer or dimer. This is in a reducing condition versus those that were treated with incomplete and that were not treated. So you can see the huge amount of interleukin-10 that is responsible for this recovery of the liver function. So our conclusions. We created a gene expression system that is both non-viral, because the majority of the systems that are used to transfect external genes and are adenovirus. And also we created or we constructed a gene expression that is specific to the liver. You can remember the image from the liver fluorescent in green. And It is because our system uses a gene promoter that is specific for the liver, which two enhancers that serve like switch on off in response to fibrotic stimulus, for instance, hypoxic signals, and that our system, that our treatment could reverse the epithelial to mesenchymal transition that is one stage previous to develop fibrosis in the hepatic stellate cells coming from a biopsy from a human. And when we tested in the on life in the mice model of cirrhosis, it was successful in revert the fibrotic process until it recovered the function and promoted the liver regeneration. I didn't show you another histological changes, but because the time, but we can assure that this supply of external interleukin-10 through this system switch on off could promote liver regeneration and in a ongoing process for cirrhosis in the mice. And we can conclude that by using the same pathogenic signals that will become the dangerous environment to develop this deadly disease, we take advantage of these same signals to promote the start on and start off of this interleukin-10 exogenous promotion to revert the damage. So the results of this investigation was a requirement of intellectual property by a patent submitted in 1999 and already sent last week to publish in Cellular and Molecular Life Sciences Journal. And this is the group of students I have to thank that were involved in this research and that are already working in the next stage that we are testing on the rheumatoid arthritis model to see the differences among the different stages of the disease. And well, this is the institution I work. It's a research center from the CONACYT centers of research. It is located in the heart of Guadalajara. You are welcome to visit. And well, I'm ready for your questions. Thank you very much.
Questions?
Anyone
Anyone?
questions?
Students first.
Okay, so my understanding, because I'm not a specialist in this topic, is that you are working still in vivo with mice, right? So my question to you is, how far are your team, for instance, to maybe test this in humans?
Well, it's an interesting question. It depends on the money and the time. Well, it's kind of difficult because the first drawback we face is in Mexico, there is not a special committee in the regulatory affair, meaning COFEPRIS, that could answer our request to validate all the data we acquire in order to proceed to human tests. So also we faced, as you can see, the patent was procured in 1999. So then we have the pandemic and we have to stop all the studies. We are already started again. So we are facing new drawbacks. For example, the money and maybe it will take less than 10 years, hope, maybe more. I don't know, to be honest. But that's the first complication we face, the money, and that there is no regulatory protocol to attend this kind of innovations.
Go ahead, yes. Like, have you tried this on another animals? It's just like
Sorry, I can understand pretty
I'm
well.
gonna repeat the question for her. Have you
Yes, yes,
tried
yes. Yes,
this
please.
protocol on other animals other than mice and not humans? Something in
Okay.
between.
Okay. Well, yes. There we use different models to test if this technology is safe by itself, not to draw the disease already installed in a animal model. We use rats and we use rabbits, but this model, the CCl4, tetrachlorine chloride, is already standardized and it's well accepted by the scientific community and also by the regulatory affairs to receive the results as valid for this disease in humans. So we think that we don't need more animals, no more models to test this technology.
Thank you.
You're welcome. Thank you very much.
Yes, Susana.
Oh, yeah, go ahead.
Well, hi, my question would be that if this treatment could be used, for example, in fatty liver, or it just has to go into the stage of fibrosis or cirrhosis.
Ah, nice question. Honestly, all of them, honestly. But at this moment, I don't have the right question, but I guess it's functional for those different stages. Well, let me point out some important information. When you become fibrotic in your liver, you already have an opportunity to recover the function. If you stop doing what you are doing, that brought you to this stage. So if you are drinking enough or you are having or taking enough soda, for example, or a high calorie diet, stop doing that. And then let your liver reverse all this damage without the need to receive such a kind of this treatment. But if you surpass this limit, then you probably require such kind of this. I mean, when you have a cirrhotic liver, there is not much options for any person to recover the function. I don't know if I answer your question.
Yes, thank you.
Okay, okay.
Good morning, Doctor. I have a question. You told that interleukin-10 is naturally produced by the liver to slow down some damages that are done naturally to the liver in our lives. But for example, if we have a genetic cause that interleukin-10 can be reduced from the liver by a natural, but in a natural way, that can cause a type of cirrhosis that is not always dependent on risk factors like drinking, and that cirrhosis may need another special treatment.
Yeah, it's probably. But first of all, you will see another kind of complications related to, for example, infectious diseases. You will see probably more recurrency on symptoms associated with flu, for instance, before you develop or any other kind of this disease in the liver or in other organs. Because if you have a mutation, for example, you will have it in the entire body, not just in the liver. But it's probably that it occurs. Yes.
Thank you.
Thank you, Doctor.
Thank you.
You're welcome.
We have one last question.
Well, I just wanted to ask, how does apoptosis like contribute or elaborate the?
Sorry, I couldn't understand it. It's back noise.
Yeah, you can repeat it.
Yes, okay, so try your best and I will try my best.
Okay, how does apoptosis contribute to the progression of the cirrhosis? Because as I know that.
Oh, wait, let me- So her question, and I'll say it in parts, is how does apoptosis contribute to the progression of cirrhosis? And then I will continue with whatever she says next.
Because I think apoptosis is like the elaboration and the self-destruction of the cell.
Because as far as she knows, apoptosis has to do with basically the death of the cell, right?
Okay, let me see if I understand. The question is how is the rate or how huge is the apoptotic event in this disease?
Yes, what's the contribution of apoptosis to the progression of the disease?
Okay. It's a good one. Well, all the questions have been good one. Okay, apoptosis and necrosis are two different processes. Apoptotic cell death is now related to inflammation, and in the opposite, necrosis, it is surrounded by inflammation. But both happen in the same tissue through this disease. And apparently there is a race between them. Which one happens first and which one become more prevalent in the organ. So this is that's the meaning of chronic lethal disease. You will be, well, not you, anyone will be losing their function because you are losing cells, but they are not removed. They remain there because the immune system that is in charge of remove any damage or dead cell cannot approach because the fibrotic extracellular matrix. So it remains like a coral. So at the end, you have lost almost the entire organ, like 90%, before you die. So that's a race or kind of between those processes. The apoptotic, that is induced by itself, by the cell, when it becomes stressed, and by the necrotic, when it is produced by the lack of oxygen and the pro-inflammatory signal surrounded. So both of those processes are part of this lethal disease along the time, in day-by-day basis. So I don't know if I answered your question.
Yes, thank you.
Okay.
Yes, thank you very much. So, very well. Thank you so much, Dr. Esquivel, for your talk. Yeah, I think it was very interesting. And what's that?
It was great having you.
Yeah, so we'll give you a final round of applause.
Thank you very much again for your kind invitation, and thank you all for being there, and stop drinking, please. Just one or two, no more, please.
Be well.
Be well, be well. Thank you very much.
Now, for all of you here, we have right now some time for lunch. We have a lunch break, and we are coming back at 2 for the next talk about edible insects, which will also be right here.
What?